Welcome to the Animal Science 105 online lab on the structure of animal tissues. My name's Martin Welby. I'm a tutor for this subject, and no doubt you'll have bumped into me by now. And I'm going to lead you through this lab today. The learning outcomes for this particular lab are listed here. By the end of the lab, you should be able to identify different tissue types, so things like epithelial tissue, which lines the inside and outside surfaces of the body, muscle tissue, glandular tissue. These are all things that we're going to look at today. And you also should be able to identify features of the tongue, so the muscle and the epithelium, connective tissue in the tongue, the lungs, the alveoli, the bronchus and bronchioles, which are the airways, blood vessels, and small intestine. We'll be looking at smooth muscle, glandular tissue, and the villi on the inner layer of the small intestine. So the technique that we use for this is called histology. Histology is the study of tissue under a microscope, and we are going to be looking at different microscope slides. So pieces of glass, and when you look at them you'll see generally a pink coloured piece of tissue. Before we get started, just like to sort of give you some idea of actually how we end up with this slide. So this particular one is a piece of tongue, and it's a bright pink colour. So what's happened here is we've taken a tongue and sliced it at about 1/10 of a millimetre thick. So to slice it that thinly, you have to solidify it first. So in this case it would have been frozen, kept frozen, and sliced while frozen. And then that very thin layer of tissue is placed on the microscope slide. When you slice any animal tissue that thinly, the tissue is colourless. So it's virtually invisible on the microscope slide. So when you look at this bright pink piece of tissue here, and when we look at it under the microscope and we see different colours, all those colours are introduced after the section is cut by adding dyes and stains to the tissue. So they're all synthetic colours. The natural colour of the tissue is completely colourless. Why do we do this? Well, form relates to function, and when you have some picture of what these tissues look like in your head, it makes understanding how they work that much easier. So we're going to run through a few different structures of the body, and when you visualise them and you have a picture in your brain, remembering the function should be a lot simpler. So the first slide that we're going to look at is this piece of skeletal muscle. It's actually from tongue of a sheep, and the bright pink colour that you can see just by holding the slide up is because all the cells in this slide are stained pink, and you'll see that the nuclei of those cells are stained purple. So just before we look at them, what are we going to be seeing? Well, we're going to be seeing muscle fibres. Now, a muscle fibre is not a typical mammalian cell. Your typical textbook mammalian cell has a cytoplasm with a single nuclei inside. A muscle fibre is actually a multinucleated cylinder. So you may wonder why I'm holding these straws. Well, actually, a muscle fibre is very similar looking to a drinking straw. It's a long cylinder, and the nuclei are all along the outside of the cylinder, and in the centre of the cylinder there are all the protein filaments that form the contracting machinery of the muscle fibre. And these muscle fibres are arranged in bundles, like a bundle of drinking straws. And the number of fibres in that bundle will reflect the strength of the muscle that it's come from. So in the tongue we will see quite small bundles, whereas if you took a piece of muscle from the leg, you'd see much larger bundles of these multinucleated cylinders. Skeletal muscle is often called striped or striated muscle, and that's because it has this characteristic striated appearance when you look under high power with the microscope. Under high power you can see lines across the cylinder, and that's because these protein filaments that run along the cylinder, where they actually overlap, they form darker bands, and you can see those under the light microscope. So that's why skeletal muscle is often referred to as striped muscle, as opposed to smooth muscle, which is another form of muscle which we will come across later on. Now these bundles of fibres are all bound together with connective tissue, so we'll see this connective tissue as well under the microscope. Before we look at this slide, I'm just going to show you a lot of these structures on a much larger scale and on a piece of material that you would be used to coming across every day. So I'll just have a look at a piece of steak, and we can see these bundles of muscle fibres and this connective tissue on a piece of steak. So here I've got some skeletal muscle that you will be a lot more familiar with. This is a couple of steaks of beef from the supermarket. But here we can see a lot of the same structures that we're talking about on a much smaller scale in the tongue. So if we look at this steak a bit closer, we can see you've got different blocks of muscle here. When you look at them, they've got this grainy appearance, and that's because the muscle fibres were running in this direction and the steak has been cut across those muscle fibres, and that's makes the steak more tender to eat, the shorter those fibres are. And you can also see how these different blocks are held together with connective tissue. So this sort of lacy material between the blocks allows the different blocks of muscle to move independently, but still holds them all together. So this is the lamb's tongue under low power. This tongue has been cut front to back and vertically, so that sort of plane. So over on the left is the tip of the tongue, the top surface is the rougher surface there, and then the smoother surface at the bottom is the underside of the tongue. Lamb's tongues, the same as ours, are rougher on top than they are on the underside. And with this slide you can basically see three different regions. You can see a darker staining pink band all the way around the outside edge, and that's the epithelial layer, smooth on the bottom, much rougher on the top. Inside of that there's a darker staining pink area which looks sort of grainy, and then in the center here there's pink areas, and you've got that the pink is in such much more sort of ribbon like structures. Now if we go to the next magnification, the reason for that differences will become clear. So here we've zoomed in on the grainy structure and the ribbon-like structure inside the tongue, and what you're basically looking at here is these ribbons of pink are muscle fibres, and you are looking at those muscle fibres from the side. They're a bit crinkled because they've contracted because the animal's dead, but they are the ribbon-like structures that are those multinucleated cylinders. All the white that you can see is just air. If you can't see any colour, it means there's nothing there. And the reason that there's so much air there is that when the thin section of the tongue is cut, the cells very quickly dry up and start to contract. So that's an artifact of the processing. That white space wouldn't be there if in the animal in real life. So these ribbon-like structures here, you're looking at the side of these muscle fibres, and then in the bottom left the structures are much more grainy looking, and that's because you have these little bundles of fibres and they've been cut this orientation, so you're looking at the end of them. So bottom left you're looking at the end of these fibres, top right you're looking at the side of them. So it's all muscle fibres, they're just different orientation. And it's that orientation of these fibres that allows the tongue to move side to side, up and down, backwards and forwards. You've got muscle fibres running in all different directions. If we go to the next magnification, the individual cylinders now start to become more apparent. So here we've got maybe 4 muscle fibres together in a little bundle, 4 here as well, single one here. This thin lacy material between the fibres is that connective tissue. Over on the bottom left here we've got the fibres cut end on, and it gives it that grainy appearance. And if we go to a further magnification, here these are the individual muscle fibres. These sort of bluey purpley coloured discs are the nuclei that are all along the outside of those fibres, and you can just about see the banding across the fibres that gives them the name of striated muscle. In your textbook you will have these pictures which are representative of what you've just seen. So the top one there shows on the far left you've got muscle fibres going from top to bottom, then you've got a bit of a space and a bunch of muscle fibres that have been cut end on, and then next to that there is some connective tissue before more muscle fibres that have been cut end on, and some more running up and down the page here. And down at the bottom we have on the left hand side muscle fibres from the side. You can clearly see the nuclei, and here muscle fibres cut transversely or cut across the end. So this next slide is a section of cat's tongue. So it's the same tissue, but this slide shows us what extra information we can get when we use a few more stains which will stain different structures, different colours. So on this slide the cells are still stained pink and the nuclei of those cells stained purple, but there are additional stains. One that stains all the connective tissue a greeny blue, and another stain which stains the keratin yellow. Now keratin is that very tough protein that is in your fingernails and in your hair, and it's what gives the upper surface of the cat's tongue that very rough feel. It has a layer of keratin on the upper surface of the tongue. So we're going to see the epidermis with this keratin on it. We're going to see skeletal muscle again. We'll also see blood vessels and the connective tissue, but this time stained a different colour. So this is the cat's tongue, and it's in the same orientation as the sheep's tongue that we looked at previously. So the tip of the tongue is over on the left hand side. the underside here nice and smooth, and the upper surface all folded and rough. And you can quite clearly see the blocks of muscle on the inner tongue, and you can see that you have some blocks running front to back which allow the tongue to be pulled in. You've got other blocks running top to bottom which flatten the tongue, and then all the way around the edge you've got blocks which are coming out of the screen, so to speak, and can allow the tongue to narrow. Between the epidermis layer, which is the dark pink staining layer on the outside, and that muscle, you can see that bluey green connective tissue, and that connects the skin to all those muscle blocks. So if we look at a bit higher magnification, and this is the upper surface, so this is one of those folds on the upper surface. You can see this dark pink staining epidermal layer. You can see all the little specks inside which are the nuclei. And when looking at those nuclei, that gives you some idea of the number of cells that are in that skin layer, because you can't see the membrane surrounding the cell. So the only way with a light microscope that you can tell how many cells are in that tissue is by the number of nuclei, and that gives you a rough indication of the number of cells. So on this fold of the tongue here, you can see this yellow stained material, and that's that keratin. So the keratin has been secreted by the cells in the epidermal layer here and is covering the upper surface of the tongue there and giving it that tough protective layer. So moving down into the body of the tongue, you've got this nice bluey green stain of the connective tissue. Again, all the little specks in there are the nuclei of the cells that are producing that connective tissue. And then down at the bottom here, we can just start to see the blocks of muscle. So to see those better, we'll move further down into the tongue. So here's at the top a bundle of muscle fibers going across the screen. You can see the individual fibers, the nuclei along the fibers, and then we've got this sort of lacy blue-green connective tissue and another block of fibers down the bottom here. In this connective tissue, this structure here is a blood vessel. So a blood vessel, particularly arteries, have smooth muscle there around the outside of them. Arteries have a much thicker wall than veins, so the wall of the artery is quite easy to see. So here we have a pink layer around a clear inside. So this is the wall of the artery and the inside there. Here is another one, and inside this artery we've still got a few red blood cells. So those darker staining objects in the artery there are red blood cells. So this is blood supplying oxygen and nutrients to all these muscle fibers in the tongue. Move right down to the bottom of the tongue now. So here you can see blocks of muscle that are coming out of the screen, so to speak. So the muscle fibers have been cut across, and it's giving it that blocky appearance. There's some fibers here running across the screen. This structure here, this big sort of long, almost sort of sausage-like structure, is a big vein on the underside of the tongue. If you look at your tongue in the mirror, and the underside of it, you've got big veins running across the underside of your tongue, and so does a cat. So this here is a vein. The wall of the vein is very much thinner. So just here you can see the vein wall, much thinner than artery would be of that size. And all these little specks in the side of that vein are again red blood cells. Over to the left here, there's a little row of 3 very small arteries. They've still retained that nice round shape because of that thick muscle wall. So when the blood pressure drops to 0 because the animal's dead, veins tend to collapse, whereas arteries hold their shape because they've got a much thicker wall. And that thicker wall is to cope with the high pressure blood that's inside, and it also means that they can contract and dilate and direct blood flow around the body. So veins, very thin walls, arteries, much thicker. And then right down the bottom here we have that epithelial layer, nice dark pink staining cells there, and a nice smooth surface which is the underside of the tongue.